Hello biology students, and this is our Macromolecule Lab preview and Macromolecule Graphic Organizer review. Again, not in class with you today, but we do need to go ahead and cover a couple of items about some things that are going to be taking place next class period. So on Wednesday afternoon. Wednesday afternoon is going to be your big Macromolecule Lab, and listen closely when I say this. This will be the most complex, detail-oriented, procedural, step-by-step, gotta focus on what you're doing in lab that you've done today, okay? It is going to be by far the most scientific, skilled lab that you've seen inside this classroom, alrighty? It's going to take focus. There are a few items that we're going to use that can be potentially hazardous to your health and hazardous to your peers, so you gotta be careful, alright? We've got to execute everything with a fair amount of caution, precaution, and just general care. Alrighty. So go to the Omaha Valley Science website, click on the Unit 3, the Chemistry of Life page. I believe I gave everybody one of these labs, said read through this thing. If not, we've got copies up in front of the smart board. Scroll down here to the Macromolecule Lab, Macromolecule ID Lab, and let's just start reading through a little bit of this information here. The purpose of the lab is to test for the presence of macromolecules in various foods. You're testing for the presence of macromolecules in various foods. These are the building blocks to life. All right, these are the organic compounds that lend themselves to assisting with everything occurring at the molecular level, structurally and functionally. So you're going to test to see where these guys can be found in different types of foods that may be present or not present in your diet. Background. The most common macromolecules, organic compounds, found in living organisms are lipids, carbs, proteins, and nucleic acids. Common foods, which often consist of plant materials or substances from animals, are also combinations of these macromolecules. Some of these compounds can be detected by taste, while others cannot. A simple carbohydrate, like sugar, you can taste that. Alrighty. However, protein, not so much. Therefore, scientists use certain tests to identify the presence of macromolecules, and it's those tests that you'll be doing in class on Wednesday in this Macromolecule Lab. Introduction. You're a scientist at the Food and Drug Administration Center for Nutrient Analysis in Atlanta, Georgia. So you work for the FDA down in the Nutrient Analysis Center in the ATL. You analyze food based on label declaration, so you look at the nutrient labels. Okay, and you make sure that it checks out that it has this percentage of carbohydrates, this percentage of sugar, this percentage of magnesium, this percentage of potassium. Tests are performed for proteins, lipids, and carbohydrates. Recently, there's been a fear of attack by a new species of undead, similar to zombies. Scientists believe that the only way to combat the attack is by feeding them a substance of high-leveled complex carbohydrates and protein, since these macromolecules appear to kill the new species of undead cells. Scientists have also found that the undead seem to thrive and grow rapidly when fed simple sugars. So, in short, to kill off this potential zombie apocalypse, we need to feed them a healthy diet. Your team is taking a break from the regular task of food label analysis in order to determine which of three substances, yogurt, milk, or oatmeal, will be the best food to fight off the invasion based on the test you will be performing today. It's up to you and your team to save the earth. So I don't know how we would get the yogurt, milk, or oatmeal to attack. I don't know how we'd use it. I don't know if we put it in a super soaker and squirt it all over the zombies. I'm not sure if we try to get them to ingest it. I don't know, but all I know is that those three substances, one of those three is going to be the best to kill off and fight off this impending zombie apocalypse. You're going to use a whole slew of materials, and some of these you're not going to recognize, but you should recognize right now a lot of your basic equipment. Test tubes, test tube racks, hot plates, beakers, glass marking pencils, pipettes, graduated cylinders, test tube holders. Some of the solutions that you should recognize: distilled water, iodine, cooking oil, oatmeal solution, gelatin solution, apple, potato, milk, and yogurt. Okay? The actual identifying solutions, so the things that you will place into these known solutions to see if they possess the macromolecules will be stuff like Benedict's solutions, iodine, biuret reagent, and What's the last one there? Sudan 3 stain. Okay. Some pre-lab questions you need to answer. What's the purpose of the distilled water as one of your test substances? Why are you going to do each one of these macromolecule tests on distilled water? Answer that for me prior to Wednesday. What's the purpose of washing the test tubes thoroughly between uses? Why is it important to start off each time with a clean, freshly washed, nothing else inside of a test tube? And finally, what macromolecule you're not testing for in this lab? And that can be found right up here. Okay, oops, sorry, not right there. But tests are performed for proteins, lipids, and carbs. Which of the four is missing? The procedure is going to be similar for each one of the tests. Okay, you're going to get 5 test tubes that are clean as can be. Label 1 cooking oil, label 1 distilled water, label 1 gelatin solution, potato solution, and apple juice. Then you need to get 5 milliliters of each one of those solutions. You'll then bring that back to your area and you will carry out the test. Okay? So for the lipid test, you'll add 5 drops of Sudan 3 stain and you'll gently shake the contents inside of the test tube. If it turns red, then lipids are present, and you record that information in the data table that's in that packet. A plus means yes, it's present. A minus means no, it's not. Take those test tubes, get those cleaned out as well as possible. Okay? Clean as can be. Then move on to the protein test. For the protein test, 5 test tubes again. You put 5 milliliters of each solution into its own test tube and make sure that it's written on that test tube what it is. That's why I have the glass marking pencils. Now you're going to take 5 drops of biuret reagent, and be careful with this stuff because it is a very strong base. Okay, so it can be a big time skin or body irritant. So be careful with that. But you can add the biuret reagent and then gently shake again the test tube. If it goes from a blue to a violet color, protein is present. If it stays the way it is, it's not present. Clean out your test tubes, get ready for the simple carb test. So you're testing for sugar, glucose here. Alright? This guy. Now this one's going to be a little interesting because you are going to again prepare each one of the test tubes as you've done for the previous 2 tests. You're going to add 10 drops of Benedict solution and you're going to get your hot plates out. You're going to go ahead and put these test tubes into a beaker that's about half full of water. Okay, and you're going to begin to heat that beaker. That way the solutions inside the test tubes can also heat up. So you're going to apply a heat source to actually make this identification take place. If you get a rusty brown color, then you have a large amount of simple sugars. That solution is chock full of sugar. If it's orange, you've got a moderate amount. Yellow to green, you've got a minimal amount. And if it stays its blue color, you got nothing. Clean all those test tubes out, prepare them for the complex carbohydrate test, which is going to simply be 5 drops of iodine, mix it up in the test tube a little bit, gently shaking it, and then see what's happened. If it turns dark blue or black, then you've got a strong amount of complex carbohydrates present. If it stays the brown color, the brown iodine color, there's no complex carbs. So, for part 1, you're testing 4 known solutions. You're testing apple juice, a gelatin solution, a potato solution, and cooking oil, and you're challenging them all against distilled water. Okay? Now, think for a second, why are you testing those 4? Okay, that's kind of an experimental group again, but really you should know just by some common everyday knowledge about which macromolecule should be heavily present in each one of those 4. Which of those macromolecules is going to be in the apple juice? Alright, so the simple sugar test, okay, for the apple juice one should give you a very good point of reference going forward into part 2. Alright, because it should test extremely well for simple sugars. So it should be a dark rusty brown after you mix it with that Benedict solution and heat it up. Gelatin solution should be a fantastic positive test for protein. Okay. Your cooking oil should be a fantastic positive test for lipids. And your potato solution should be a great test for complex carbohydrates or starches. Okay, so you're testing all of those to see what a very, very strong positive test would look like as a point of reference going forward. And then you're also going to have some mixed results with a few of the tests as well. So you're basically building your background reference knowledge here for part 2, which is all about identifying whether milk, oatmeal, or yogurt is going to be the best solution to fight off the zombie apocalypse. So reading through procedure, part 2 right here, I will give you milk, yogurt, or oatmeal, and you'll go ahead and perform each one of those tests on it. So you only need 4 test tubes for this, 5 milliliters of whatever solution I give you in each test tube, and then one of the test tubes will go through the lipid test, another one through the complex carb, another one through the simple carb, and another one through the protein. Record your results in the table. I'll also have everybody put the results onto a document online, and then we're good to go. Okay? Clean everything up. Everything is left as clean as can be and placed back appropriately. That's going to be big for this. All right, it's going to be big for this lab. Okay? And then prior to the next class period, you'll need to answer all the analysis and conclusion questions. And that could be something that you might have time at the end of class to complete as well. Okay? So you got this big time lab on Wednesday, and it's looking like we are not having school on Friday. So we'll have a test adjustment, and it would be, I'm going to assume, Monday when we see you back here. Okay, I don't know if we're going to switch the days around or not, but we'll get it figured out. But we still have to get lesson 4 in after the Macromolecule Lab and then our test. Okay, that's it for the Macromolecule Lab. As for the graphic organizer, which can also be found online, you need to watch the video and make sure that this information is filled in. Okay, this will help you out greatly in completion of the lab, building your vocabulary for this unit. And also, we are going to see these 4 macromolecules rear their ugly heads through Unit 4, 5, and 6. We're going to see these a lot. They're going to be a major topic and theme of discussion here going forward. So get this bad boy filled out using the macromolecule video for Lesson 3. You can also use your textbook. A lot of great information to fill this out can be found in your textbook in Section 3, somewhere between pages 28 and 42. Okay? Ready through this lab. Be prepared for Wednesday. Adios.